Well, let's start. So we're in for a real treat today. I know many of us are very excited to hear Andy's presentation. Just real quick background, Andy graduated both bachelor's and doctoral degree, University of British Columbia. He did a postdoc just south in San Diego under David Kleinfeld, and he is currently holds an appointment at both the Seattle Children's Research Institute and the University of Washington in Seattle. So David's going to be talking about capillary function in the brain and some of his optical techniques to study that. We're all very excited to hear you, Andy. Thank you.
Thank you so much for the invitation, and I'm really excited to share some of our published and unpublished results, especially with this audience that's very well versed in optical techniques and optical imaging. I'd love to get some feedback. So thank you so much for the introduction. So our lab is very interested in cerebral vasculature. We use primarily multi-photon imaging to study the physiology of the smallest blood vessels in the brain. Not only just to observe, but also to use light to manipulate the cells and begin to do cause and effect type experiments to understand how blood flow is regulated within the brain. And we primarily use rodent models to do this research. So I'd like to start the talk by saying that, in our brains, in the adult human brain, there's 400 miles of total vasculature. And when I first started studying the cerebral vasculature, I said, that's impossible. How could that be? That's basically from Irvine to San Francisco for you guys. And for me, it would be from here to Portland and back again. That's quite a bit of vasculature. And it's really hard to imagine how much of that length could be packed into a brain. But with the work that we've been doing and also the very careful measurements that we've been doing at the smaller scale, I'm beginning to believe that is actually true. A huge amount of vascular length has to be perfused by blood cells in order to maintain moment-to-moment brain function. So perhaps the easiest way to start thinking about this is using clinical imaging techniques that allow us to first have a view into how perfusion in the brain actually works. The idea being that blood is supplied through the carotid arteries, goes into the internal carotid artery, as you can see here. Now, this is a Ctu angiogram. So basically a dye has been injected into the bloodstream, and then you're just tracking the front of that dye as it courses through an adult's brain. And you can see that dye move, and this is now showing the middle cerebral artery, one of the largest arteries in the brain that supply most of the parietal cortex. And then a few seconds later, as you take these CT angiograms, you can see that it becomes more of a diffuse type of label. And this is where it's entering what some clinicians call the capillary blush, where all of the dye is now entering something that's below the resolution that can be achieved with the imaging technique. And it just looks like this diffuse haze as all the contrast agent is entering the capillary networks of the brain. And then a couple seconds later, all that dye now enters a completely different vascular network, the venous vascular network that you can see is draining it away and backed out through the sagittal sinus. So that 400 miles of vasculature, which I'll explain more in detail later, has been traversed in a matter of 5 seconds by the collective flow of blood within the brain. Now, some really beautiful work from Henri de Verneuil. He was a vascular physiologist in France, did a lot of his work back in the mid 80s. And some of the most beautiful diagrams that he generated were from studies where he injected India ink into cadavers. And that India ink could perfuse through the tissue, through the vasculature, and then he would be able to extract the tissue and take a look at it and just take beautiful pictures and to draw out what he thought was going on. This is one of the more restricted views of the surface of the cerebral cortex, surface of the brain. You can already see very large arteries and smaller sort of arteries branching off of those large ones, and then little loops that are forming anastomotic connections to form redundancy within the vascular network. There's some veins in here as well. Every once in a while, you can see some of these penetrating arterioles sort of branch off, and they seem like they end, but they don't actually end. They dive into the brain tissue. So if you could just take a cut and look at that tissue on the side, you would see something like this, where one of the penetrating vessels now go into the cortex, supply the deep tissues, and ramify off into these smaller branches called precapillary arterioles and into this very dense plexus of capillaries. So you can see that most of the vascular length, albeit these vessels are very small in diameter, lie below the surface of the brain in the form of capillaries. Now, this is our version of divergent noise experiment in the rat. We also do this, most of our work in the mouse. So any rodent is good for us to study. And what we've done is we've injected fluorescent gelatin dye. So the idea is during perfusion, we inject this and then we cool down the animal, take the brain out, and then it labels all the vasculature in the same way, creating a vascular cast. Again, you can see the branches of the middle cerebral artery. Those are the thinner branches that are coming off here, but also the larger veins that drain from the same territory back to the sagittal sinus, which normally would be here, but was removed when we extracted the brain tissue. Now, with two-photon imaging, we want to study these vascular networks in the brain of a living animal. So we've got to actually remove the skin, the skull. In the case of a rat, we have to peel back the dura mater, part of the meninges. In the mouse, we can leave the dura intact because it's thin enough for us to image through. But in creating these cranial windows, you begin to get a different type of view, something a little bit more focused on the overall networks. So here again is the surface of the brain. This is a composite of many images collected by two-photon microscopy from a living adult rat. I pseudo-colored the vasculature to help you guys understand what is an artery and what's a vein. So again, these are the branches of the middle cerebral artery and the big branches of the veins. These are the small branches coming off that penetrate into the tissue. So a lot of similarities with the human vascular network. But again, we're only scratching the surface like in the human brain. What we want to do is zoom in even further to understand what the other scale of complexity looks like at the level of the capillaries. And so now if we zoom in even further, this is more like the typical field of view that we would be working with to understand blood flow dynamics in a rodent brain. This is about a cubic millimeter of mouse cortex now, very similar to a rat. And you can see one of these penetrating arterioles branch off, and then they dive into the brain. So this is compressed into a two-dimensional image, but what would be about a cubic millimeter of tissue. You can see this little haze around it. This is called the Virchow-Robin space that's filled by CSF. You can see these little branches coming off the precapillary arterioles, and then it enters a labyrinth of capillaries that we frankly know very little about, and how blood flow is actually regulated at scale, even though it is the most predominant thing that you would see when you do this type of imaging. All that blood has to percolate through that sieve-like network, find its way through it, and then come out and converge out through the ascending venules and back into the veins on the surface of the brain. The capillaries go all the way up, right up to the surface of the brain, all the way down to the white matter, which are at the bottom. This is a side view of just a small part of this, and you can see that the densities change across the different layers of cerebral cortex. Now, we've done very careful calculations of this, but also other groups like David Kleinfeld's group, where what we've done is segmented the entire vascular network in this cubic millimeter of mouse cortex and measured how long the vascular length is. And there's a meter of total vascular length. So that's pretty unbelievable on its own right. That's so much vascular length that's packed into literally a pinhead-sized piece of tissue. And these are how calculations of hundreds of miles of vasculature existing in the human brain. This is where it comes from, if you were to scale the volume up to the size of a human brain. And you can also appreciate it from looking at these types of images that most of that vascular length is composed of capillaries. Now, two-photon imaging isn't just a structural form of imaging. You can get dynamics. You can get movies at optically sectioning through the tissue. This is after a fluorescent dye has been injected into the bloodstream, fluorescein dextran. It's a high molecular weight dye that doesn't leak out into the tissue, doesn't pass the blood-brain barrier, but it also isn't picked up by any of the blood cells. So the blood cells look like little shadows that are moving through a fluorescent sea, and you can track the movement of those cells in order to calculate blood flow at the level of single capillaries within the brain. Also labeled in this animal are astrocytes using a dye called sulfurrhodamine 101. And you can see how the astrocyte cell bodies are apparent, but also their processes form part of the blood-brain barrier wall. Oops. So this is in all of our physiology textbooks. It's very clear that arteries are designed to regulate blood flow in the brain. They're muscularized. They have these ring-like smooth muscle cells around them, and they enable very dynamic control of cerebral blood flow from the second to second scale. However, I also just shown you that all that blood that goes through the arteries has to eventually pass through an expansive capillary system. And blood cells are actually larger than the diameter of a capillary lumen. A blood cell being 6 microns on average in diameter, a capillary lumen being more about 3 to 4 microns. So the blood cell actually needs to distort, and the capillary lumen needs to expand a little bit to get the blood to pass. And that's a very important process because that resistance slows down the blood in the right way to extract oxygen from the blood in order to supply to the tissue. You can also then imagine that even very small changes, perhaps the ones that we can barely resolve with optical imaging, can have a profound influence on the way that blood flows through the brain. So the overarching question then is capillary diameter even regulated to influence blood flow? And that's something that has been very difficult to get at, not only because of the spatial scale that you need to image at, but also because there's an inherent elephant in the room. If you want to study capillaries, you have to dissociate it from what's going on in the arteries. We already know that arteries regulate blood flow, but if you want to study things at the capillary level, there will always be contamination of arterial effects from upstream. So how do you dissociate these vascular compartments and understand them separately? So we tried to tackle that question using various types of optical approaches. And one of the things that we really needed to understand was what actually lives on the outside of the wall of a capillary. We know that concentric ring-like smooth muscle cells are on arteries, but by the time you get down to the capillary, the mural cell, or the cell that covers the outside of the endothelium, the tube of the vessel, is called a parasite, and they look completely different from what a smooth muscle cell looks like. They have these protruding cell bodies, sometimes called bump on a log cell bodies, and very long processes that don't completely cover the endothelium. So they don't really look like they're structurally equipped to do the job, to regulate blood flow. Oftentimes we look at these, we study parasites, histologically, and including using electron microscopy. And this is a schematic of what you might see during this type of work, where the parasite sits on the outside of the endothelium. It has peg and socket interactions with the endothelium, a bump on a log cell body that protrudes outward, and the entire cell is encased within an extracellular matrix called the basement membrane. So this question of whether or not parasites and by extension blood flow in the capillaries are independently, autonomously regulated is not a new question. It's actually over 100 years old. In fact, the person that discovered parasites, Rouget, posed this idea. He said that these cells were, they seem to be in the right place, and they seem to be positioned in a way that might be able to control blood flow down one branch or another. Of course, he didn't have the tools to really study that question. And over the course of then, this idea got forgotten for a while, but then in the 70s and the 80s, different studies were performed in peripheral tissues like cremaster muscle, retina, frog tissues, and they all sort of pointed at the idea that there might be some regulation of blood flow. But there were also a good steady stream of papers that suggested the opposite. And as you can see now, even in our era, there are papers that are published in Nature, Neuron. These are high impact journals that don't seem to agree on whether or not parasites can regulate blood flow. So I'm not going to dig into the details of why this controversy exists too much, because I want to talk more about what we've done to try to move forward. But I do want to say that this is a really important question. And the reason why it's important is because there's no lack of translational relevance to studying these cells. There's no controversy that in the aged brain, and also in the context of Alzheimer's disease, that parasites are lost at an accelerated rate. And it's not uncommon to have reports of 25 to 50% reduction in parasite number in Alzheimer's disease patients. And currently, that parasite loss has been most strongly linked to changes in the blood-brain barrier, which can be also queried within the same histological postmortem tissues. But what effect that parasite loss has on the way that blood flows through the brain is a physiological question, and that's very difficult to get at without the use of preclinical models and better imaging approaches in humans. Another thing that I want to talk about is that the histology has revealed other types of vascular features that are very curious. String capillaries, they're basically collapsed, non-patent or non-flowing basement membrane tubes within the brain. And these are very common in the Alzheimer's disease brain as well, and we don't really know why they arise, but we can imagine that they probably are not very good for the way that blood flows through the brain. So in order to get at studying this in a preclinical model, we had to develop the right tools to do the job. So the first question was, how do we even find parasites in the brain, and how do we define what they are in order to clarify exactly where we are studying within the vascular network? So one of the things we did was clear brain tissue from animals that had transgenic labeling of all mural cells. So PDGFR beta is a marker of all mural cells. We can cross these mice with reporters of TD tomato in order to get really bright labeling of all mural cells. This is one of the penetrating arterioles from these animals. And what I'm trying to show you in this movie is that alpha smooth muscle actin, which is shown in green here, or yellow with the co-label, which is a very potent contractile protein, doesn't go all the way into the capillary bed. It's certainly on the arteries, and it's on some of the precapillary arteries. But by the time you get down to the capillary zone, there's no contractile protein there. And that's one of the reasons why people believe that perhaps parasites aren't able to regulate blood flow, because of the lack of this critical protein. So what we do then is we define parasites and the capillary network as these zones that lack alpha smooth muscle actin, which is the key question. We know that things that express alpha smooth muscle actin are very good at contracting, and they regulate blood flow in a dynamic way. There's no question about that. But what about all the rest of the other stuff? What about these vessels downstream that do not express high levels of alpha smooth muscle actin? Do they regulate blood flow? And that's how we make this definition. We also are able to define it carefully by saying, if we count this many branch orders away from an artery, we know at some point we enter that zone. And so that way we can very carefully specify where we are within this otherwise very complicated vascular network. From these tissue clearing studies where we did alpha smooth muscle actin labeling, we also now can appreciate that most of that vascular network doesn't have alpha smooth muscle actin. If you take the total vascular length and just say how much of it actually has contractile protein expression, 4%. So a minority of the total vascular length actually has any of this. So most of it is alpha smooth muscle actin negative, and therefore, by our definition, of the capillary bed. So capillary parasites are associated with more than 90% of the total vascular length in the cortex. So what are the morphological characteristics of capillaries? And one of the things that we could do with these transgenic animals is do sparse labeling of the cells in order to get a better picture of what they actually look like. And I think that this was lacking before we actually started looking at these cells. And a combination of histology and high resolution confocal imaging allowed us to see that these cells, they had very small cell bodies, but most of their cell body was, most of their length was actually in their processes, where they would reach out over hundreds of microns of length. They would often touch multiple capillary segments. Sometimes they would have these helical-like processes that would wrap around the outside of the endothelial lumen. And sometimes they were just very thin strands that run along the outside of the endothelium. You can go in even closer using super resolution confocal microscopy, and you begin to see more substructure. Here you can see these sort of serrated edges on the outside of the parasite. And now we know from a combination of this type of work and also three-dimensional EM imaging that each of these little serrated edges coming off are actually like claws that are grappling onto the endothelium underneath, allowing them to have a mechanical foothold onto the endothelium and presumably be able to exert some tension along the vascular wall too, which is what we're going to get into. Also, if you guys have any questions along the way, please do just unmute and ask your questions as we go. So with these transgenic animals, we could also create cranial windows and study the parasites in vivo. And in combination with the intravenous dye that I had showed you earlier called Fascidal. So the parasites are in red, the intravenous dye is in green. And you can see that the cell bodies are present. You can see their long processes, but this also revealed that by looking at these cells in a three-dimensional network, that they don't always cover the vascular endothelium. In fact, sometimes they have small gaps in between their territories, but they didn't like to occupy each other's territory. They were very territorial in nature. They had their own domain. And every once in a while, you'd come across a gap where you could see where the two processes of neighboring parasites would interact with each other. And this was kind of interesting because it suggested to us that these cells were plastic in their ability to grow. Like in some cases, one parasite would grow a little bit and the other one would retract away. It's not very fast, but these things are sort of twitching a little bit. But it then opened the question of whether or not these cells were mobile or had the ability to change their shapes. And so we did another experiment, again, using optical approaches. So the lesson here from this was that the parasites form a cellular chain along the capillary bed. They don't overlap and they negotiate their territories with each other. The experiment that we had thought of from seeing this type of data is that we should try to optically ablate parasites in an otherwise normal network without affecting anything upstream or downstream. Let's try to mimic what's going on to some extent in the human brain and see what happens. And so this was not too hard to do because, two-photon imaging, when we teach our students not to use too much laser power in order not to change the very thing that you're interested in looking at, you can also then tell them once in a while to up the power and just try to blast something. And it's kind of fun to be able to do these experiments and just take, a line scan across a cell body of one of the cells and then ablate it entirely. You can see here we've ablated one of the parasites, not affecting any of the other parasites within the network. And we've done a lot of controls as well to make sure that this laser ablation itself is not creating massive amounts of damage. Rather, we're just creating enough power to thermally ablate the cell and not affect any of the other cells around it. The control that we did was called an off-target control. We want to know that the laser itself isn't creating a lot of the physiological effects that we're looking at. The laser itself, of course, is not benign. The heat that it generates can create changes in vascular diameter or disrupt the neurovascular unit. So what we did was we took the same type of laser scan and just moved it a little bit away from the cell body in order to have a comparison of just what the laser itself was doing. So here's one of the experiments. We ablated a parasite and it revealed its territory quite clearly. And then what we saw was over the course of a couple of weeks after this, that the neighboring cells became plastic and they started to remodel their processes and grow back into this area and in order to maintain coverage of the endothelium. So after parasite ablation, processes of neighboring parasites show augmented growth and seem programmed to regain coverage. And that coverage must be very important. Also in the course of these studies, just to keep things more focused, I'm not going to go into the detail on this, but we didn't see any evidence of the proliferation of these cells or migration of new parasites coming in from other territories. And also a little bit, heretical with the field, which is well established where parasites, when they're lost, they can disrupt the blood-brain barrier. We didn't see any evidence of blood-brain barrier disruption here. We didn't see any leakage of the intravascular dye. And we also have bred these animals with microglial transgenic mice that are very potent immune surveillance of vascular damage. And we don't see the microglial cells interacting much with the vasculature either. So then what is the consequence of losing parasite coverage in the capillary network? And so the most consistent thing that we saw in these experiments was that when that period of parasite loss was happening before the neighbors start to grow back in, the underlying endothelium looks like it has dilated. You can see in this example here where there's parasite contact, but then as soon as the parasite contact is lost, the vessel dilates. And then when the neighboring cells grow back into that territory, then the tone comes back. So it really does seem that parasites, at least their contact is important for maintaining the diameter and the flow of the capillary network. And here's some composite data to show you what this looks like, where the dilation is only present when the parasites are not in contact with the underlying endothelium. Of course, diameter doesn't tell you everything. We want to know what's going on with flow. And as I mentioned earlier, that the shadows moving through the fluorescent C within our angiogram allows us to track the movement of blood cells. Here, you could take one capillary and just track the shadow in a very fast movie. But we're wasting a lot of time scanning dark areas that have no information. So what we can also do is just take a line scan through the middle. And these iterative line scans can be then appended on top of each other to create a cascade image that looks like this. Essentially a ticker tape of information about how fast the blood cells are moving and how many blood cells have moved through in that given time that you were scanning. We get speed by looking at the change in distance, which is the x-axis here, and then over the change in time, which is the instantaneous speed of that one blood cell that was moving through. But also the number of cells. Each of these shadows is essentially one cell moving through, and we can count the number of these and call it the flux, or the cells per second. So if you put all that information together, the ablation and our ability to measure blood flow at the level of single capillaries, we can test the question of what happens to blood flow when we lose parasite coverage. So here's an on-target scan. We ablated the cell. The vessel dilates. The ticker tape clearly tells us there are many more shadows moving through, and therefore the amount of blood flow going through that capillary has increased quite a bit. And just to be thorough, we want to show that the sham irradiation doesn't have the same overall outcome. And again, here, where we moved the scan to an off-target region, you can see that there's no increase in blood flow. The composite data across many capillaries and many animals shows that capillary diameter increases with capillary parasite ablation, but also the flux increases as well. Okay, so we've established by looking at this that the ablation of capillary parasites can lead to vasodilation and aberrantly high blood flow levels. But Alzheimer's disease, as I posed at the very beginning, is a disease of the aged brain, and everything that we've done so far is in the young adult brain. So we started to age animals and study if the same consequence was occurring when these animals were about 2 years old, which is corresponding to about 70 to 80 human years. This is, let me just frame this a little bit. So these are the two groups that we looked at. Now, everything shown here in red and green is basically capillary length that has been uncovered by ablating a series of parasites, probably 2 or 3 parasites in a row to get much more of the capillary bed uncovered. The green area is what has been regained by remodeling of neighboring cells over the course of 21 days. And then the red areas are the regions that remain uncovered despite that period of time for regrowth. And you can see that in the aged animals, the ability to recover and regain tone is much more limited. A way that we can look at it here is the amount of the capillary length that's uncovered as a function of time. And you can see that it's just a slower growth and there's just more of the capillary bed uncovered. And even in some animals where we tracked over more than a month to see if eventually things would become covered again by the parasites, we see that there's still gaps that are formed. So essentially you're pushing the limits of the remodeling process because there's not enough parasite growth. And we attribute this to a reduction in the speed at which these neighboring cells are growing their processes outward. But interestingly, this dilation that we saw during the period where the parasites were not present was exacerbated in the aged animal. So here you can see that they're about maybe about half a micron or more dilated compared to the adult animals. Not really sure exactly why there's an exacerbation and dilation, but it could be something to do with the basement membrane integrity or just the vessels themselves become more flaccid as aging occurs. Here's one of the more drastic examples. You can see that this capillary is nearly tripled in size and its diameter, but the growth of neighboring cells can bring that tone back. And I mentioned earlier that some of these regions never regain any coverage, and those regions remain dilated for the entire period of time. So essentially in the age brain, if you lose parasites, you cannot get parasites to cover as efficiently, and you have these road bumps, dilated regions spread throughout the capillary network. So after parasite loss, aberrant capillary dilation is exacerbated in the aged brain, and further, aging diminishes parasite plasticity and prolongs the loss of parasite coverage and thus prolongs capillary dilation. So you might be asking, well, how could this actually occur? Where does this extra blood come from if you don't change anything upstream or downstream? So let me just illustrate this thought a little bit. What we've done is we've ablated a parasite somewhere embedded within the deep parts of the capillary network, but we haven't fundamentally changed anything upstream or downstream, which begs the question of where that extra blood came from. And the answer to that question comes from understanding that not all capillaries are the same at baseline levels. Some capillaries are very small in diameter and then flow more slowly under basal conditions, and some of them are large and they flow many more cells under basal conditions. So there's an incredible heterogeneity, not only in the diameter of capillaries, but also in their flow. And we don't really understand why this heterogeneity exists, but it's clear that the system is designed in a way that'll make sure that blood coming through the artery is distributed properly through the entire capillary network. And if we manipulate these diameters, we might be changing the way that blood is flowing through in a negative way. So the system has sort of reached like an equilibrium. And what happens when you start to mess with it? And so then the question that we posed is, well, let's imagine there, for the sake of simplicity, there's two types of parasites, one on small diameter capillaries and one on large diameter capillaries. And we ablated, by having this mixture of different parasite types within the network, that you create sort of thoroughfare channels or patterns of flow that are consistent from day to day. Perhaps, adapted to the metabolic demands of the tissue and what the tissue needs. But if we were to ablate the parasite on one of these small diameter capillaries, then you could simply be just redistributing blood or stealing blood away from areas that would normally need more blood. So does this actually happen? And so in order to test this idea, we had to do a different type of experiment where we couldn't measure many capillaries over, or a few capillaries over many animals, but rather we had to measure many, many capillaries in a single animal. And this real difficult experiment was done by one of my graduate students, where she ablated 3 parasites in a row. This region was uncovered, and then measured as many capillaries as she could around the region of ablation, not only looking at the flux of the cells, but also the directionality of flow, that she could understand where things were coming from and where things were going out. And so she categorized the capillaries into 2 groups, ones that were not covered, so therefore dilated, and ones that remained covered in the periphery. And you can see here in this data that in some of the uncovered neighboring cells, there actually is a reduction in blood flow. And in the non-covered areas where there's a dilation, there's an increase in blood flow. So we're just redistributing blood and we're pushing them in different patterns. Is that good or bad? That's a good question, right? So the basis of why this is happening is that at certain bifurcations within the capillary network, decisions have to be made about how much is blood to send down one daughter branch versus another daughter branch. And by deleting parasites on one side, you essentially just dilate that branch and allow a path of least resistance for all the blood to flow down. And the other daughter branch, which would be normally receiving some blood, has now reduced in blood flow, or I'll show you in a little bit, has stopped blood flow altogether. Now, these are very painstaking experiments to do in vivo. So we coupled with a group in Switzerland to do some modeling in silico studies, where they had taken full reconstructions of capillary networks from mouse brain and used this to understand the distribution of blood flow through a network. And we asked them to mimic the experiment that we were doing in vivo, essentially taking about 14 capillary segments, essentially what we would be uncovering with 3 parasite ablations in a row, and say, what happens to the surrounding capillaries in terms of their flow within this capillary network? And what we found was that many more of the capillaries surrounding these tissues were changing, either increasing or decreasing above a threshold of 10% of their baseline level. And 2/3 of these increased in blood flow, but consistently 1/3 of them decreased in blood flow as well. And interestingly, the amount of dilation that we exerted on these very focal capillaries made a difference as well. So if we dilated the capillaries to an extent that we saw in the aged brain, it would have reverberating effects throughout the capillary network in a much broader way than a small dilation, of course. And we pushed the system even more, doing something a little bit aphysiological, but just to see what would happen. And essentially, the more you dilate, the more you recruit disturbances in the rest of the capillary network. And there was always some vessels that were decreasing in blood flow in addition to the ones that were increasing in blood flow. So what does this mean? Well, when we go back to the animal and look at our experiments more carefully, there's a type of reduction in blood flow that is so extreme that it really doesn't take much measurement to see. And it's essentially a loss in blood flow altogether. So the blood cells stop flowing. And it looks like there's no shadows moving through. And sometimes there's a cell that's sort of stalled within the capillary. And this is one of the examples where there's this one precapillary vessel branches into 3 capillaries. The one in the middle loses parasite coverage, it dilates, and now is a shunt that has taken all of the blood. The one on the left side no longer flows because there's no longer a need, there's no longer pressure pushing it down that way. And this one over here has regressed away because somewhere before our imaging session, that has stopped flowing and the endothelium has regressed away. And it looks an awful lot like a string capillary, where if you could stain for the basement membrane, you probably would see it's still there. It's just there's nothing, there's no luminal space in between. So in 10 of the 14 experiments that we did, we saw stalls somewhere surrounding the capillary net the capillaries that we had dilated, and then in a subset of those we saw regressions. And I just wanted to emphasize that we're not doing very profound changes. We're ablating 2-3 capillaries at a time, and we're already seeing these types of effects. And I can only imagine what's happening when you lose 25 to 50% of the parasites within the brain over the course of a disease like Alzheimer's. So there's got to be a very profound vascular component to Alzheimer's. So just to summarize this first portion, the parasites maintain basal capillary tone. What I've shown you there is that if you ablate these cells, you have a dilation, augmented blood flow. And this comes at the consequence of maldistribution of blood flow within the capillary networks. And then the capillary network remodels itself in order to remove some of the non-flowing networks, parts of the network, and they become something that looks like string capillaries. And interestingly, these string capillaries do not start flowing again, even after parasite coverage comes back and things seem to be restabilized. We've lost some of the capillary segments permanently. So what I've shown you is that capillary parasites are necessary to maintain tone of a capillary bed, but I haven't really shown you whether or not they're actually sufficient to do the job. And implicit in the question of whether or not these cells actively regulate blood flow at the level of the capillary bed. And that's a different experiment, and another one that you can leverage optical imaging to get at in a cause and effect sort of way. Now, this animal line that we have labels all cells, all mural cells, including smooth muscle cells. So we couldn't simply just, manipulate all the cells at the same time, because you would not be able to dissociate arteries from capillaries. But using optics and optogenetics, we could then now target our optical activation of parasites in a cell-specific manner. So most of you probably know what optogenetics is, but for those of you that don't, I'll quickly just mention it. It's the ectopic expression of a light-gated ion channel in the cell of choice, in this case, in all mural cells. And the idea is that normally you would apply blue light onto the cell, and it would open up the channel and it would influx cations, sodium and a little bit of calcium, and it would cause the cell to depolarize. And in the case of smooth muscle cells, that depolarization will lead to contraction of the smooth muscle and reduction in the diameter of the vessel. But we don't know what this would do to parasites. To get at this in a sort of a spatially restricted way, we had to do two-photon optogenetics. Now, it turned out that 800 nanometer light was a pretty good means to manipulate the optogenetics probe within the parasites in a way that didn't affect anything upstream or downstream. And we really restricted the way that the line scans were being projected onto these images. So to kind of dig in a little bit deeper, to be really careful that we weren't activating any smooth muscle cells upstream, we would track, move really far away from the penetrating arteriole, 5 branch orders away, where we knew that we were definitely in the capillary bed. And then in that region, we would make sure there was nothing above or below that would, confound the results of our local stimulation. And then we applied a line scan that did 3 things. One, the wavelength was sufficient to activate channel rhodopsin. We would scan across the vessel to measure the diameter of the vessel, and we would scan once down the middle to measure the blood cell flux and the velocity of blood cells. This is what the line scan trajectory looked like. And this is what one of the line scans would look like. So I basically took this and just sort of cut it and spread it along. You can see that it crosses the diameter of the capillary 5 times, goes once through the middle, and allows us to track blood cell movement. And then even within the 60 seconds of scanning of this channel rhodopsin expressing mouse, you can see that the diameters of the capillaries have changed and the number of blood cells moving through has reduced. So that suggested to us that these cells were able to be stimulated. We wanted to be really careful, though, to make sure that the light itself wasn't the reason why we were getting vasoconstriction. So we had control animals. These animals expressed only YFP without channel rhodopsin. And we stimulated them in a way that we used the same laser power at the same depths. We established a depth power relationship for the laser power. And applied that approach to both of these genotypes of animals. And this is the composite data that we got from these experiments, showing that the channel rhodopsin animals constrict more at the capillary level. These are all capillaries. And then also as they constrict, the blood cell velocity is reduced. And interestingly, the blood cell velocity reduces after the constriction. So this suggests to us that this constriction has to be a local effect that reduces then blood flow passage through that part, rather than the alternate hypothesis is that we're stimulating something that propagates upstream to an artery, and then the artery upstream is contracting. In which case, if that was the case, if that was going on, then you would expect blood flow to reduce before or much earlier than this effect. And then finally, the number of blood cells moving through has reduced as well. So this is pretty good evidence to us that we could specifically drive these cells to contract if you stimulate them enough. So capillary parasites can be optogenetically stimulated to contract and decrease blood flow. And now that we have this assay, we asked, well, is there a way that we can test whether some of the drugs that we know to work on smooth muscle cells and arteries upstream also have a functional effect on the capillary network? That would be very interesting to know, because then we would know that the mechanism of action of some of these drugs might be much broader than we think. One of the drugs that we were interested in using is Fascidal. Fascidal is a Rho kinase inhibitor. In a nutshell, what it does is it dilates smooth muscle cells by inhibiting actomyosin contractile machinery. Predominantly, it is used in China and Japan as a treatment for subarachnoid hemorrhage, vasospasm after subarachnoid hemorrhage. This is one of the Ctu angiograms from this type of work, where you inject Fascidal intravenously, and it basically helps to open up the artery and improve perfusion to the brain. It is not yet FDA approved here to be used in the US, but there is evidence that it is has salutary effects in stroke, and it is fairly tolerable and has pretty good safety, clinical safety profile. And so the question was, what about the parasites? What is happening to the capillary bed? So we took Fascidal and we applied it to the surface of the brain to get around some of the broad systemic effects of dilating blood vessels. And we said, does the topical application of Fascidal have any effect on our ability to cell-specifically contract parasites? And we saw a dose-dependent reduction in the ability to contract the parasite down with Fascidal, and also a dose-dependent ability to reduce blood flow. So basically, we could block optogenetic stimulation and contraction of the parasites. So Fascidal might actually be working at the capillary level in many of these patients as well. And the other thing that we could do with optogenetics is use our probe like a circuit board. Let's compare the kinetics of contraction at the capillary level with the kinetics that we know occur at the arteriolar level and also at the venous level. And what we found was not surprising, that arteries upstream are very good at contracting, and they were very, very robust when we optogenetically stimulated them. And the parasites could contract, but they were sort of slow and gradual. So they had a much slower kinetic to them. And some of these transitional vessels in between also were a bit slower than these larger upstream arterioles. And then downstream in the veins, we saw very little contraction at all. So it seems to me that most of the dynamic regulation of blood flow is happening at the level of arteries, and more the basal slow regulation of blood flow is happening at the level of the parasites through the capillary bed. This is also to show that the absolute change in diameter that can be exerted by optogenetic contraction is big in the arteries, larger in these sort of transitional parasites called ensheathing parasites, and then in the predominant type of true parasite in the capillary bed that I've been talking mostly about, it's just a very slow, gradual contraction. So summary number 2 is that capillary parasites are sufficient and necessary to alter capillary tone and reduce blood cell flow. And putting this information together, I've shown you now that optogenetic activation of capillary parasites in a cell-specific manner can reduce blood flow in a slow means compared to other mural cell types. And then earlier I mentioned that ablation can lead to prolonged dilation, augmentation of flow. So we think that these parasites sitting here in this middle zone are very important for basal capillary tone and blood flow resistance. And they might actually be regulating that heterogeneity of blood flow, basically creating a level of pressure along different parts of the capillary network to properly distribute blood to all parts of the brain. And that heterogeneity is very important and something that we want to dig deeper into in our next set of studies. I've also shown you that both of these pathways, this pathway here can be inhibited by Fascidal. But that this pathway over here has a reparative strategy too, with the growth of parasites that we're also looking into pharmacologically to see if we can get them to grow more quickly. But we do think that in that period of time where there's no capillary parasite coverage, that there's a maldistribution of blood flow, and this perhaps could be exacerbated in the context of Alzheimer's disease. Let's see, I think we're okay on time. We're okay on time, right? Okay. I just wanted one more section to go through really quickly. This is new unpublished stuff that I wanted to get some feedback on. So what I've shown you is that using, fancy but non-physiological, optical approaches, we can study the parasites in more detail, but we also want to know what's happening naturally over the course of aging and also in diseases. And this is something a little bit more difficult to get at because inherently it's an observational type of experiment where you've got to just sit and watch and see what happens to the parasites. And so we asked, well, what's happening in the normal aging brain? This is the foundation for all subsequent studies of disease mechanisms that are associated with age. And there is some research that's been done showing that there's a loss of parasite coverage just with normal healthy brain aging. And we wanted to understand what this does to blood flow. But something was not quite right. When we looked at these aged animals, compared them to the adult animals, and looked at capillary length capillary junctions, and also the abundance of parasites within these networks, we didn't see any difference. And that kind of bothered us a little bit because it suggests to us that this parasite loss isn't naturally happening at all in the animals that we were looking at. But then we also realized that maybe this is a region-specific type of effect. So all of these studies that we have been doing in these manipulations and were done in the upper layers of cortex, probably within the first 250 microns of cortex. But I also showed you in some of those earlier images that the blood flow has to penetrate all the way down to the base of the cortex and into the subcortical white matter. And we don't know much about how blood flows in that system at all. So we had to use a different type of imaging. We call this deep two-photon imaging. This has been spearheaded by some other groups, and we kind of adopted their strategy. The idea being that we want to push deeper than conventional two-photon imaging using redshifted intravenous dyes, longer wavelengths that don't scatter as much, and improved optical lenses to allow transmission in the infrared range. And effectively, what we've been able to do is double the imaging depth that we can achieve in both adult and aged mice. Here's an example of what this type of data looks like. It's very, very clear. This is a large swath of brain tissue, a montage of multiple regions, and you can see that we can image quite deep into the brain tissue. And also, I wanted to point out this large vein right here. So by the time we got down to the depths of these regions that we had never imaged before, we started to realize that there were a predominance of veins, and that we had not seen in the upper layers of cortex. And so when we reconstructed these types of data and looked at the types of blood vessels within it, what we noticed was that there were these very, very large veins called principal cortical veins that penetrate all the way down into the deepest parts of the gray matter and into the white matter underlying it. It's got a Christmas tree-like structure. Most of its arborization is in the deep layers of cortex, and all these other veins that I'm showing right now are the other veins that we thought we understood very well in the upper layers of cortex. In fact, they don't even penetrate down to the deep layers of cortex. There's a bottleneck in perfusion where all that blood going deep into the brain has to come out through a single output through these principal cortical veins. The other interesting thing is that there's no shortage of arteries that get blood flow down to that depth. There's a ring of many, many penetrating arterioles, and all the blood flow, however, has to come out through a single output. It's always good to know that what you're studying in the mouse brain is, has a correlate to what we see in the human brain. And going back to de Vernoy's work, some of his beautiful schematics that he had drawn showed exactly what we saw, principal cortical veins. And you could see he put it right in the middle of his schematic diagram, emphasizing how important these veins were. He also showed that these veins were surrounded by a ring of penetrating arterioles. And just to provide a little bit more context, these principal cortical veins are still, they're perfusing cortex and some of the sort of underlying superficial white matter tracts of the U fibers of the human brain, which in the mouse brain, the sort of the similar type of white matter tract was what we would call the corpus callosum. But they serve a similar purpose in that this tract is important for communicating information across different regions of cortex. So then what we could do is segment out and understand these these venous networks that were adorned with capillaries across different layers of cortex. And this is what these networks look like. This is just one of the branches coming off of the principal cortical vein. And all the vessels shown here in gray are capillary sized vessels. And so the predominant type of vessel within this massive draining network are all capillaries. And we also did some 3-photon imaging with Jack Waters and Kevin Takasaki at the Allen Institute. 3-photon imaging similarly allows very deep penetration into the tissue, but it also offers another thing, a third harmonic generation signal that tells us where myelinated fibers are. And we could use this information to figure out where these principal cortical vein network branches were starting to intersect with the gray and white matter interface. So we learned from doing these studies that these deep branches were actually flanking both the white matter and the gray matter, and that we could actually measure blood flow this deep, and particularly a blood flow in the actual white matter of the living animal's brain. So to cut to the chase, we did a lot of vasodynamic measurements, and what we found was that the diameter changes associated with age didn't happen in the upper layers of cortex at all. They happened deep within the brain, far away from the perfusion source. Also, the changes in the densities of the capillaries happened deep within the brain. Not only that, the reductions in blood flow that we were expecting to see with brain aging didn't happen in the upper layers. They were actually associated with the deep layers of the brain. And in this case, with the red blood cell velocity, we actually saw an increase in blood flow, which has been reported by some groups doing similar types of imaging, where they said aging is associated with increased blood flow. Well, that's not the case. What we think is probably happening is that there's a redistribution of blood, an impedance of blood flow in deep layers, and therefore blood has to exit through upper layers of cortex. And so very, very important to be able to image through the entire depth of the cortex, and hopefully even deeper, to be able to really understand what's going on with natural age-related vascular phenomena. So capillaries most distant to the arterial perfusion source, or the watershed regions, exhibit selective vasoconstriction, reduced blood flow, and reduced density. And we think, although we didn't actually look yet, that the aberrant contraction of parasites and parasite loss might be involved in these selective deep tissue changes. So with that, in the last 1 minute, I'm going to just put this all together for you guys. What I'm trying to get at here is that there's a blood flow impairment that we know occurs in aging and Alzheimer's disease. And we think that Alzheimer's, a parasite contraction, or the aberrant contraction of the parasites, similar to what we've seen when we stimulate them with optogenetics, is occurring when there's these types of pathologies. Other groups have also shown this, and some of the different vasoactive mediators that can create this have also been revealed. But the new data that we've added here is that at some point, these parasites have to die off in order to, manifest the reduction in parasite coverage that has been reported in pathology. And when that happens, there's a maldistribution of blood flow, there's a stalling of blood flow, and there's a regression of blood vessels that leads to a fundamental change in the architecture of the capillary network. And that feeds back into this loop, and the whole thing just keeps on going. And so we need to figure out a way to stop this positive feedback loop. And there's two things we need to do. We need to control parasite contractility, and perhaps Fascidal or something similar to that might be useful in that respect. And we're also trying to assay different types of signaling pathways that would allow us to harness parasite endothelial contact. So to get the parasites to grow faster, or maybe proliferate in order to maintain parasite coverage. So with that, I'd like to thank you very much for your attention. I want to say that this work, none of it was done by me. I was just a cheerleader for these three really talented individuals. André Bertiam did all the parasite ablation studies. David Hartman did most of the optogenetic studies, and Stefan Stemenkovic, a postdoc in my lab currently, did all the deep imaging work. These are collaborators, our various funding sources, and I'd like to thank you very much for your attention.
Thank you, Andy. That was wonderful. Very, very interesting work. We have a couple questions that people have put into the chat. I can read them. Or David Cribbs, are parasites lost in older Alzheimer's disease transgenic mouse models relative to aged match controls?
Yes, yeah. So the mouse models are highly varied. There's many, many different types. So I'll speak specifically to one type that we're very interested in. It's called the TG Sweetie model, and it has certain mutations in it that allow more of a vascular associated accumulation of amyloid plaque. And in that model, we've seen with imaging and also histology that there is natural age-related parasite loss, and it's exacerbated beyond what we see Actually, we haven't actually shown in our lab that parasites are lost with normal aging, but we can definitely see in this model compared to, a littermate controls that there is a reduction in the number of parasites. And we're along the lines of trying to understand what natural parasite loss or naturally occurring in the context of a disease model looks like, and whether our optical manipulations are at all mimicking, recapitulating what is normally happening in the brain. I think that's an important part. So that's sort of next on our agenda to get at.
Okay. Amoko Watanabe asks, what are the environmental consequences of dilated capillaries by the loss of parasites? That's the first question. Then there's a kind of a follow-up. How would planting parasites in Alzheimer's disease mice help to mitigate the pathology?
Yeah, no, that's a great question. And by environmental consequence, I'm assuming like, what happens to the tissue surrounding the capillaries, including the, the function of neurons, the oxygenation of the tissue, the function of astrocytes. We've been almost wholly focused on the blood vessels right now. And what we want to do now is start to bring in neuronal calcium imaging, Two-Photon imaging of oxygen content using a new probe called Oxy42P that actually reports the partial pressure of oxygen in the tissue. And that goes along our goal of testing the hypothesis that these parasite dilations, they aren't necessarily good. In fact, they're creating pockets of tissue that are hypooxygenated or hypoperfused and therefore bad for brain function. And so we haven't clearly shown that yet, but we're trying to develop the tools to get closer to that. Transplanting parasite, that's a great question as well. There have been some studies saying that you could, put like some stem cell alike, early progenitors of parasites, and they can actually take on parasite functions and migrate to the vascular wall and essentially do the job of a parasite. It's very early days, I think. I saw one paper on this, but I do know that there are some groups actually at University of Southern California, Vetsa Slokovich's lab, that is really deep into that question. And I think it's a really good one. We just personally haven't done any work on that. I could read the questions too if needed. Yeah. So Misha asks, could fibrinogen be related? Could fibrinogen be related to affect capillary diameter as it is a blood coagulant protein? It's asking, since parasite degeneration leads to dysfunction of white matter circulation and blood-brain barrier disruption. Yes, interesting. We haven't seen a lot of things that look like overt pathology like you would expect to see in terms of blood-brain barrier breakdown or coagulation of the vessels. We've done a lot of research looking at photothrombotic occlusion of small vessels. I know what it looks like when a capillary has been coagulated or like in a photothrombotic means, and it's not pretty. The vessels will break down, the vessels leak, bad things happen. But I was surprised actually when parasite loss was being generated in our model system. It seemed pretty normal, like the brain could take it pretty well in that respect, but let's see, at least in a local sense, right? And it wasn't until we really thought about blood flow dynamics that we realized that something was important to be understood in that respect. But we were very surprised that we didn't see any clotting, blood-brain barrier breakdown. And this is at odds a little bit with some of the published literature, I think, which is what you're alluding to, that parasite loss leads to blood-brain barrier breakdown, leads to circulatory failure, it leads to loss of neurovascular coupling. It's just very, very bad. And I would say that there are two things to answer in that respect, and it relates to the models. One type of model that was used to study parasites in the past was one called basically a congenital loss of platelet-derived growth factor. Basically, the cells, the parasites never really migrated during development to make proper capillary networks, and the vascular wall is abnormal through the entire course of the animal's life. So we think that that is not an ideal model, because it really, the presumption is with Alzheimer's disease is that we're all sort of normal to begin with, and then something bad happens and an accelerated worsening of cerebral vascular function occurs. And I think that model, it doesn't really get at it, because we want to see an age-related effect after normal development. And the other thing that has been done has been genetic deletion of parasites in a much broader fashion, using things like diphtheria toxin, where you can remove 50% of the parasites within a week or something, in the adult time frame after normal development, which is getting better because there's no developmental component to it. But in that particular experiment, you're losing parasites so rapidly and so quickly that it's really hard to dissociate what the primary effects of parasite loss are from the secondary effects. If a parasite was to contract and, down, then you would get coagulation, and then you would get stroke, and then that would lead to blood-brain barrier breakdown. But whether or not the parasites immediately had an effect on the blood-brain barrier is very hard to tease apart when it gets that complicated. So we're sort of on the other end of the spectrum for this type of research, where we're just sort of finessing it to see what these cells are doing at a very fundamental level. That's a great question. Richard Robinson asks, have you any data or interest in effects of mild traumatic injury on parasite capillary relationship? Yeah, I'm interested in a lot of things, but I cannot do a lot of things at the same time. I have to focus quite a bit. I'm interested in anything that has a relation to disease-related parasite loss, disease-related capillary dysfunction. There are some other groups here in Seattle that are very good at traumatic brain injury models, and we've been talking a little bit, but we haven't really picked up much on it. With respect to the data that's out there, parasite loss and parasite migration away from the vasculature is pretty well established to occur in injury models as well. And that's not stroke. Stroke, it's been shown in stroke as well, but in models like CCI models and such, there have been some studies to show that the parasites die and migrate away from the vasculature.
Other questions for Andy? Andy, that was really very fantastic, extremely interesting. Very much appreciate you giving a presentation to all of us today.
Well, thank you so much for the invitation, and for the great questions and the great feedback. Yeah, it's great to be able to share the results, and hope you guys all stay happy and healthy.
Okay. Oh,
one more question.
Yeah, another question.
I wanted to know if incorporating dopamine via multiphoton imaging could help stabilize the function of vessels through acting on parasites and endothelial cells, since I have heard low levels of dopamine can be associated with memory loss. Yes, I have not thought about that particular interaction yet. That's a great question. It's so good that I don't have an answer for it. I'm sorry. Very interesting though. But I will say one thing though. The parasites might have receptors for a lot of neurotransmitters. And some of the recent single cell transcriptomic data that's coming out, not only from human vasculature, but also rodent vasculature, could be something of interest to mine through to determine whether or not dopamine or other neurotransmitters have a direct sort of receptor interaction with the parasite that could do a host of intracellular things, which we know very little about. I have to say. But that's a fantastic question and probably something worth diving into in the future.
May I have a quick, maybe last question too? I was wondering if you are familiar with Chris.
Oh, I'm sorry. I think you cut out for a little bit. I couldn't hear the last part.
I know. Can you hear me now?
Yes, I can.
I was wondering if you're familiar with Chris Shu's work from Cornell, who was able to, it's more on the technology side of the two-photon microscopy. He was able to push the penetration depth down to 1.6 millimeters. Is there anything more to see within that between 1 and 1.6 millimeters?
Oh, yeah.
Okay.
It's like the wild, wild west right now. I mean, the deeper you can go, the more interesting it gets. I mean, if you think about what's below what we've been able to image, I mean, if depending on where you are in the anterior posterior axis, you can image the hippocampus. But at 1.6, you could get to pretty deep into the pyramidal layer of the hippocampus, probably. And in the hippocampal vasculature, there has been some work that was done from Catherine Hall's lab, where albeit they had to decorticate, remove the cortex to do their studies. If you could do it without removing the cortex, that would be great. What they've shown there is that the vascular density and the parasite density is much lower in the hippocampus, meaning that their threshold for resilience to parasite loss must be lower to begin with. So that in the context of Alzheimer's disease and aging is a transformative technology, I would say, what Chris is doing there. If you were to move a little bit more anterior, you would access the fibers of descending pyramidal neurons going into the subcortex of the striatum, perhaps even imaging the white matter tracts of the internal capsule. If you talk to clinicians, I'm sure Mark Fisher, if he's listening, he would agree with me that white matter is where most of the action is happening in terms of these diseases. We see white matter degeneration is predominant within age-related dementias. And so being able to image at the cellular level and be able to understand the cellular level contributions to white matter degeneration would be huge. It would open so many doors for what we do in our models.
Is it just his group using this very long wavelength? I think he's using something like 1.7 microns.
Yeah, I think so. I think that they're definitely on the forefront of pushing this, and we're sort of following in the background, trying to use some of the technology and the advances they've had. So I can only speak mainly from, I don't know how many, there are a couple groups I know of that I've been interacting with that do 3-photon and have any interest in blood vessels. One of them is Jack Waters, who I'm collaborating with at the Allen Institute, but also Prakash Kara at University of Minnesota is very interested as well. Chris Schaefer, who works closely with Chris Shu. Sava Sakadzik. I don't know if they do actually with 3-photon, but anyways, it's technology driven and it's technology limited. Not a lot of labs have those expensive equipment or the expertise to do that very well. And but hopefully it'll get easier. And so our application with just regular 2-photon imaging and long wavelength dyes has already opened up a set of doors for us that we can do. So hopefully the threshold for that technology becomes more accessible, is reduced. Yeah,
that's great. This is great work. Impressive.
Thank you.
Actually, I was going to ask a question right up to your last slide. How do you close the loop? And that was
how do you close the loop?
Yeah, and you did it. So
yeah. Oh, good. Thanks. Yeah. No, I should just leave it open ended. I was like, what do you think? And so yeah, I think it's it's pretty clear that there's got to be a feedback loop here. So we're moving more toward just trying to figure out how to manipulate the system, manipulate what's happening. Well, understanding what's happening normally in these disease models and then trying to manipulate it.
Mm-hmm.
Yeah.
Great. Thank you again.
Well, thank you so much, everyone.
Okay.
Thank you.
Bye-bye.
Bye.